Good morning, dear students. Whether my voice is audible to everyone?
Good morning, ma'am.
Okay, fine. Yesterday, I assigned some work for Soumya Naral, Chaitra, Lohit, and one more is Samath. Samath is not here, I guess. Is he here? No, Samath did not log in yet. So, Chaitra, Lohit, and Soumya Naral sent me the questions. Okay. Now, what is the thing is, fine. So from today, as from yesterday, we started regarding the special, the applied biology, the specific topics on biology with principles of inheritance and variance. Okay. So from this week onwards, we will be dealing with such concepts only. So in this, from this week, we are going to concentrate more on practicing by ourselves. No doubt you will be testing that, you will be having the test every evening. From that, you will get to know your marks and your preparation level. From today, will be having two tasks, guys. So the first task is I will be assigning certain students to pick up some question from the lesson which I discussed today. Tomorrow, you have to message me the discussions. Please don't answer for the questions. Lohit, I said you to only prepare for two questions. Okay, please don't answer everyone about the questions. And guys, ನಾನು ಯಾರಿಗೆ ಕ್ವಶನ್ಸ್ ನೀವು ಕಳಿಸಿ ಅಂತ ಹೇಳಿರ್ತೀನೋ ಅವರು ಎವ್ರಿ1 ಚಾಟ್ ಬಾಕ್ಸ್ ಅಲ್ಲಿ ಎವ್ರಿ1 ಅವ್ರಿಗೆ ಕಳಿಸೋದಿಲ್ಲ. ಯು ಹ್ಯಾವ್ ಟು ಸೆಂಡ್ ಟು ಮೀ. ಮೈ ನೇಮ್ ಇಸ್ ನಿಧಿಶ್ರೀ. ಓಕೆ, ದ ಕ್ವಶನ್ ಶುಡ್ ಬಿ ಸೆಂಡ್ ಟು ಮೀ. ಅದರ್ವೈಸ್ ನೀವು ಕಳಿಸಿದ್ರೆ ಅವ್ರು ಆನ್ಸರ್ ಮಾಡ್ತಾ ಹೋಗ್ತಾರೆ. ಫೈನ್. ಸೊ ಪ್ಲೀಸ್ ಡೋಂಟ್ ಡೂ ದಟ್. The question should be sent to only me, not to everyone. Fine. Okay. Then, just a minute. Then after this, from today, you will be having that task. The second task is last 10 or 5 minutes, we are going to discuss 5-star questions. Bio 5-star questions on the topics which we covered today or else which we covered yesterday. ಎಸ್ಟರ್ಡೇ ಕವರ್ ಮಾಡಿರೋ ಟಾಪಿಕ್ ಬಗ್ಗೆ ಆದ್ರೂ ನಾನು ಕೇಳ್ಬಹುದು, ಇವತ್ ಕವರ್ ಮಾಡಿರೋ ಟಾಪಿಕ್ ಬಗ್ಗೆನೂ ಕೇಳ್ಬಹುದು. ಇದ್ರಲ್ಲಿ ಏನ್ ಸ್ಪೆಷಲ್ ಅಂತ ಅಂದ್ರೆ, ಇಟ್ ಈಸ್ ಅ 5-star bio. 5-star bio. The name is 5-star bio. Why 5-star bio means the main 5 questions will be asked from the well-known publications. CET, NEET ಗಂತ ತುಂಬಾ ಒಳ್ಳೆ ಬುಕ್ಸ್ ಇವೆ ಮಾರ್ಕೆಟ್ ಅಲ್ಲಿ, ತುಂಬಾ ಒಳ್ಳೆ ಪಬ್ಲಿಕೇಶನ್ಸ್ ಗಳಿವೆ. ಸೊ ಎಲ್ಲಾ ಪಬ್ಲಿಕೇಶನ್ಸ್ ನ ನೀವು ತಂದು ಓದಕ್ಕಾಗಲ್ಲ. So what I am doing is, I have certain books, I have certain publication issued by the company. So ಆ publications ಅಲ್ಲಿ ಇರೋ ಕ್ವಶನ್ ಅನ್ನ I will be presenting. Okay, we are referring that publication. I will be selecting the questions from that book and I will be presenting to you. ಅದಕ್ಕೆ 5-star ಅಂತ ಯಾಕೆ ಕೊಟ್ಟಿದ್ದೀನಿ ಅಂದ್ರೆ, ನಾನು ಲಾಸ್ಟ್ 5 minutes ಅಲ್ಲಿ 5 questions ಕೇಳ್ತೀನಿ. ಒಂದು ಕ್ವಶನ್ ಗೆ 5 seconds ಆನ್ಸರ್ ಇರುತ್ತೆ. That is, I will start 5, 4, 3, 2, 1. I will start 5, 4, 3, 2, 1. Okay. Then when I start 5, 4, 3, 2, 1, ಈ 3, 2, 1 ಅನ್ನೋ ಅಷ್ಟೊತ್ತಿಗೆ ಯಾರು ಫಸ್ಟ್ ಆನ್ಸರ್ ಮಾಡಿರ್ತಾರೋ, so those people, I mean that student will be a winner of the day. Fine. Frequent ಆಗಿ ಯಾರು ವಿನ್ನರ್ ಆಗ್ತಾರೋ, that student will be getting a gift from me. Fine. Okay. And I request all the students for whom I will be assigning the work. Okay, please don't message in the chat box. That too to everyone. Please message to me only. Fine. Let us proceed with the students' question now. Soumya Naral messaged me first. Her question is, all of you listen, please. Her question regarding principle of inheritance and variation. Her answer. Pratibha, mute yourself. Okay. So your answer should be in the chat box. No one should speak about students' question. But avrigu reply maadu hagilla. So Soumya Nara asked the question, in Morgan's experiment, F2 ratio deviated very significantly from 9:3:3:1 ratio. This is due to, ಯಾಕೆ deviate ಆಯ್ತು ratio ಅಂತ ಕೇಳಿದ್ದಾರೆ. Option A, recombination. Option B, independent assortment. Option C, segregation. Option D, linkage. Don't answer through mic, answer through chat. Apurva, Preeti, Angel.
ಮ್ಯಾಮ್ ಆನ್ಸರ್ ಹೆಂಗ್ ಮಾಡ್ಬೇಕು ಮ್ಯಾಮ್?
ಚಾಟ್ ಅಲ್ಲಿ ಮಾಡ್ಬೇಕು, ಮೆಸೇಜ್ ಅಲ್ಲಿ. ಓಕೆ, ನಾನು ಯಾರಿಗೆ ಅಸೈನ್ ಮಾಡಿರ್ತೀನೋ ಇವತ್ತು ಅವ್ರಷ್ಟೇ ನಾಳೆ ಕ್ವಶನ್ಸ್ ತಗೊಂಬನ್ನಿ. ಎಲ್ಲರೂ ಕ್ವಶನ್ಸ್ ತಗೊಂಬರೋವಾಗಿಲ್ಲ. It creates mess in chat box. Saundarya R, option B. Preeti, option C. Viresh, option B. Fine. Be fast. Chetana, Prema, Nuhu Sayyed, Oppo. Okay, Kanchana Raj, Pavitra. Most of them voted for option B. Few of them selected option C. Yes, Soumya Narar, who is correct? Which option is correct? Can you explain?
Yes ma'am, option D, linkage.
Option D, linkage. Can you explain what is linkage is? Anyone who want to define linkage? Okay. Option D, linkage is a right answer. Angel answered option D. Then only few of them answered as option D. Next, Lohit. Lohit, are you here? Lohit Kumar.
Yes, ma'am.
Yeah, please do ask your question. 1 minute. I will just check your question. So can you send your question once again, Lohit? because most of them messaged here. Okay, Chandru, they will be sending you the question. Lohit, please send the question. Okay, let us proceed with the question of Lohit Kumar. The number of contrasting characters studied by Mendel for his experiment was. The number of contrasting characters studied by Mendel for his experiment was option A 14, option B 4, option C 2, option D 7.
Ma'am, once again repeat question.
The number
Option
of
D, ma'am.
contrasting characters ಮೆಂಡಲ್ ಅವರು ಎಷ್ಟು ಕಾಂಟ್ರಾಸ್ಟಿಂಗ್ ಕ್ಯಾರೆಕ್ಟರ್ಸ್ ಅನ್ನ ಲಿಸ್ಟ್ ಔಟ್ ಮಾಡ್ತಾರೆ 7, 14, 10 and 9 something.
Ma'am, 7, ma'am.
7, ma'am.
7,
Okay.
ma'am.
Almost all of you answered it as 7. Very easy question and very quick response. Good. Now, Lohit, second question is multiple alleles control inheritance of. Multiple alleles anta ide, hauda? So it controls the inheritance of option A sickle cell anemia, option B color blindness, option C phenylketonuria, option D blood group. Angel answered as option B, Apurva option D, Vidya option D, Preeti option D, Kavita option D, Saundarya, Pavitra, most of them answering as option D, blood group. Okay. Yes, Lohit, is it correct or wrong?
Correct, ma'am.
Blood group is a right answer. Now, Chaitra. Fine. The Chaitra's question is, what is called pleiotropism? Pleiotropism andre enu? Very good question. Phenomenon of multiple effects of simple gene. Option B, phenomenon of multiple effects of 2 gene. Option C, phenomenon of multiple effects of multiple gene. Option D, all of the above. Pleiotropism andre enu anta kelidare. Option A alli enide phenomenon of multiple effects of simple gene. Please chat your answers, don't speak up. And option B, phenomenon of multiple effects of 2 gene anta. I got 3 new messages. Lakshmi Odayar option C, Oppo A16 option C, Kanchana Raj option C, Veeresh option C, Pavitra, Saundarya D, Saumyanaral option C, Prema option C, Duti option C. Yes, Chaitra, which is the correct answer?
Option A, ma'am.
Option A.
Phenomenon
Can
of multiple effects of simple gene.
ಮಲ್ಟಿಪಲ್ ಎಫೆಕ್ಟ್ ಅಂದ್ರೆ ಒಂದೇ ಜೀನು ಡಿಫ್ರೆಂಟ್ ಫಿನೋಟೈಪಿಕ್ ಎಫೆಕ್ಟ್ಸ್ ನ ಒಂದಕ್ಕಿಂತ ಹೆಚ್ಚು ಫಿನೋಟೈಪಿಕ್ ಎಫೆಕ್ಟ್ಸ್ ನ ತೋರಿಸ್ತಾ ಇರತ್ತೆ. ಅದಕ್ಕೆ ಪ್ಲಿಯೋಟ್ರೋಪಿಸಂ ಅಂತ ಕರೀತಾರೆ. ನೆಕ್ಸ್ಟ್, Saumya Naral Singh, second question goes like this. Okay, it is 2. Okay, statement 1. I will ask you this question later. Yakandre, this question is too long. If I explain them through the mic, they will not able to answer, Saumya.
Okay, ma'am.
Okay, fine. Then, this is regarding the questions from students and certain suggestions. ನಾನು ಯಾವ ಸ್ಟೂಡೆಂಟ್ಸ್ಗೆ ಅಸೈನ್ ಮಾಡಿರ್ತೀನೋ ದೇ ಶುಡ್ ಬಿ ಓನ್ಲಿ ಆಸ್ಕಿಂಗ್ 2 questions. 2 questions ಅನ್ನ ಕೇಳಬೇಕು. ಯಾವಾಗ ಅಂತ ಅಂದ್ರೆ ಡೋಂಟ್ ಮೆಸೇಜ್ ಮೀ ಡೈರೆಕ್ಟ್ಲಿ once the session starts. ನಾನು ಹೇಳ್ತೀನಿ, ಫಾರ್ ಎಕ್ಸಾಂಪಲ್, ಇಫ್ ಐ assign the work for Kanchana Raj tomorrow, so Kanchana Raj should send me the question when I ask. Okay, then you guys should answer so that you guys can also read the question properly in the chat box. Artha aita? Understood all of you?
Yes, ma'am.
Okay. Let us proceed for today's concept regarding molecular basis of inheritance. Just a minute. Molecular basis of inheritance. So, first we have regarding the discovery of DNA, Watson and Crick model. They used X-ray crystallography. X-ray crystallography. DNA, chemical language that is produced in all cells, direct development of traits or characters. Then, After that, DNA discovery Frederick Griffith ಅನ್ನೋರು ಸ್ಟ್ರೆಪ್ಟೋಕಾಕಸ್ ನಿಮೋನಿಯಾ ಬ್ಯಾಕ್ಟೀರಿಯಾದಲ್ಲಿ ಸ್ಟಡಿ ಮಾಡ್ತಾರೆ. So he found out two strains. One disease causing, which is called pathogenic. One is non-pathogenic. Killed pathogenic bacteria with heat, mixed killed with non-pathogenic living. So avaga some living became pathogenic, transformed, inherited in all offspring. Experiment na explain ಮಾಡ್ತಾ ಇದ್ದಾರೆ. So I'm sorry. Transformation. He explained a process which is called as transformation. What do you mean by transformation? It is changing genotype and phenotype due to assimilation of external DNA by a cell. Genotype ಮತ್ತೆ phenotype ಎರಡು ಕೂಡ ಚೇಂಜ್ ಆಗುತ್ತೆ. ಯಾಕೆ? Assimilation of external DNA, foreign DNA, ಹೊರಗಿನ DNA ಬಂದು ಇನ್ಸರ್ಟ್ ಆಗೋದ್ರಿಂದ there will be change in genotype and phenotype. So this is regarding discovery of Frederick Griffith. Next, fine. So experiment ಹೇಳ್ತಾರೆ living S cells ಅಂದ್ರೆ pathogenic cells. So ಇದನ್ನ inject ಮಾಡಿದಾಗ mouse dies. Living R cells non-pathogenic. So ಅವಾಗ mouse healthy ಇರುತ್ತೆ. Then heat killed S cells ಅಂದ್ರೆ pathogenic strains ಅನ್ನ ಅವರು heat ಮಾಡಿ ಕೊಟ್ಟಾಗ again heat killed control S cells. Mouse is healthy. Then after that, mixture of heat killed S cells and living R cells anna kottaga mouse dies. So from this he will come to know DNA is a genetic material. Then genetic info, DNA or a protein. Fine. Samarth. Now he sent the question. Okay. So Samarth question is ನಾನು ಯಾರಿಗೆ ಹೇಳ್ತೀನೋ ಅವರಷ್ಟೇ ಕ್ವಶನ್ಸ್ ಅನ್ನ ತಗೊಂಡ್ ಬನ್ನಿ. ಮೋಸ್ಟ್ ಆಫ್ ಯು ಆರ್ ಸಿಂಪ್ಲಿ ಆಸ್ಕಿಂಗ್ ಸೆಂಡಿಂಗ್ ಮೀ ದ ಕ್ವಶನ್. ಯಾರಿಗೆ ಅಸೈನ್ ಮಾಡಿರ್ತೀನೋ ಅವ್ರು ಕ್ವಶನ್ ತಗೊಂಡ್ ಬನ್ನಿ. ಓಕೆ ಫೈನ್, ಲೆಟ್ ಅಸ್ ಡಿಸ್ಕಸ್ ಇಟ್ ಲೇಟರ್. ದೆನ್ Kanchana Raj, can you read out the slide please?
Yes, ma'am. Genetic information, DNA or protein. Bacteria phase. Viruses that infect bacteria. Viruses are the simple and non-living. DNA or RNA and protein code required a host to reproduce. Hershey and Chase used radioactive sulfur tag protein, used radioactive phosphorus tag DNA. Centrifuged infected bacteria, found phosphorus in the bacteria. Therefore, genetic information is equal to DNA, not protein.
So here DNA genetic information na correct ಆಗಿ ಮಾಡ್ಕೊಳ್ಳಕ್ಕೆ whether DNA is a protein ಅನ್ನೋ question ಬಂದಾಗ Hershey and Chase concluded or performed certain experiment. They used radioactive sulfur, radioactive phosphorus which tag protein, radioactive phosphorus tag DNA. ಅದನ್ನ ಸೆಂಟ್ರಿಫ್ಯೂಗ್ ಮಾಡಿದಾಗ ಇನ್ಫೆಕ್ಟೆಡ್ ಬ್ಯಾಕ್ಟೀರಿಯಾನ they found phosphorus in the bacteria. So phosphorus tagged DNA. Therefore ಏನ್ ಗೊತ್ತಾಗತ್ತೆ, DNA is not a protein, it is the genetic material. Fine. So let us proceed to the questions of molecular basis of inheritance. Okay, the first question of today is who coined the term nuclei? I will not give the options. Answer. Nuclei ಅನ್ನೋದನ್ನ ಯಾರು coin ಮಾಡಿದ್ರು? Because that the question slide got missed. So I have an answer slide.
Frederick
Mischer. Okay. What about others? Chandru Waggar, Frederick. Okay. What about others? Okay. The right answer is Frederick Mischer. He credited with the discovery of DNA in 1869. He identified it as an acidic substance in the nucleus. As he found it in the nucleus, he gave the name nuclei. Good. Second question goes like this. Purines found both in DNA and RNA are option A, cytosine and thymine, Option B, adenine and thymine. Option C, adenine and guanine. Option D, guanine and cytosine.
Option C, ma'am.
Option C,
The
ma'am.
true answer, option C. Oppo A16, option C. Rama, Chandra, Pavitra, Lakshmi, Veeresh, Prema, Yes, Harshita, Chaitra, Veeresh, Nuhu Sayyed, Kumar, Saundarya, Gayatri. Most of them are not answering. Why? Kangita, Sangeeta, Parvati, Aishwarya. Don't make me to take your name. Present idru answer ಮಾಡ್ತಿಲ್ಲ. Veerappa, Vidya. Okay. Most of them voted for option C, that is adenine and guanine. Okay. The names who are not answering, the names will be recorded. Meeting recorded irutte, gottagate yaar answer madalla anta. Parents will be called. Okay. Then. So the right answer is option C, adenine and guanine. Okay. The term nuclein for the genetic material was used by, already discussed, it is used by Mitchell. Yeah. The 3' to 4' phosphodiester linkages. The 3' to 5' phosphodiester linkages inside a polynucleotide chain. I repeat, inside a polynucleotide chain serve to join. ಏನಾಗತ್ತೆ ಏನನ್ನ ಜಾಯಿನ್ ಮಾಡ್ತವೆ ಅವು ಆಪ್ಷನ್ ಎ ಒನ್ ನ್ಯೂಕ್ಲಿಯೋಸೈಡ್ ಟು ಅನದರ್ ನ್ಯೂಕ್ಲಿಯೋಸೈಡ್ ಆಪ್ಷನ್ ಬಿ ನ್ಯೂಕ್ಲಿಯೋಟೈಡ್ ಟು ಅನದರ್ ನ್ಯೂಕ್ಲಿಯೋಟೈಡ್ ಆಪ್ಷನ್ ಸಿ nitrogenous base with pentose sugar. Option D, DNA strand with the other DNA strand. Who is going to answer first? Kaveri.
Option B, ma'am.
Option B, ma'am. One nucleotide with another
One
nucleotide.
nucleotide with another nucleotide.
Option B, most of them answering as option B. Kanchana, Soumya, Lovit answered through Mike. Sinchana, Veeresh, Veerappa, Asha Patil, Gayatri, Sangeeta. Okay, Nuhu Sayyed, Parvati. Most of you are answering it correctly. Option 2, that is one nucleotide with another nucleotide, is the right answer. Okay. Can anyone read out the slide?
Nucleotides are building blocks of nucleic acid like DNA and RNA is formed of 3 components like pentose sugar. It is the 5-carbon sugar that always occurs in pentose form. Nitrogenous base. It is the N-containing heterocyclic base ring compound in 2 types of nucleic acid. There are 5 types of nitrogen bases: adenine, guanine, cytosine, uracil, and thymine. Phosphate group. It is formed of phosphoric group and it is present in both DNA and RNA in nucleic acid of phosphate meiotic link. The tertiary carbon of one sugar of one nucleotide to the 5 carbon of succeeding nucleotide. So 3 to 5 phosphodic linkage inside a polynucleotide chain. So way to join one nucleotide with another nucleotide chain. Nucleic acid or polynucleotide, like the polymeric compound of one nucleotide. A nucleic acid which contains deoxyribose sugar is called as DNA, while the one which contains ribose sugar is called RNA.
Very good. So why I make you to read the slide? Because you will get the other points also here. It helps you. Thank you, Kanchana Raj. Number of base pairs in human chromosomes are option A, 3 into 10 raised to 9. Option B, 3 into 10 raised to 7. Option C, 6 into 10 raised to 8. Option D, 6 into 10 raised to 7.
Option A, ma'am.
Option A, ma'am.
Okay.
3 into 10 raised to 9.
Parvati, Prema. Most of them voting for option B. Kumar, Rajeshwari, Asha Patil, Madhumita, Sangeeta, Kavita. They voted for option A. Varsha, Saundarya, Nuhu Sayyed. Fine. Most of you answered for option A. Few of you attempted it as option B. So the right answer is 3 into 10 raised to 9. Option A is the right answer. The next one is a molecule that can act as a genetic material must fulfill the triads given below. ಈ ಒಂದು ಸಬ್ಸ್ಟೆನ್ಸ್ ಅಥವಾ ಮಟೀರಿಯಲ್ ಜೆನೆಟಿಕ್ ಮಟೀರಿಯಲ್ ಆಗಿ ಆಕ್ಟ್ ಮಾಡ್ಬೇಕಾದ್ರೆ ಇಟ್ ಹ್ಯಾಸ್ ಸರ್ಟನ್ ಕ್ವಾಲಿಟೀಸ್. ಈ 4 ಆಪ್ಷನ್ಸ್ ಅಲ್ಲಿ ಯಾವ ಕ್ವಾಲಿಟೀಸ್ ಇರಲ್ಲ ಜೆನೆಟಿಕ್ ಮಟೀರಿಯಲ್ಗೆ, ಅಂದ್ರೆ ಇಟ್ ಇಸ್ ನಾಟ್ ನೆಸೆಸರಿ ಅಂತ. Which quality is not necessary? It should be able to express itself in the form of Mendelian character. It should be able to generate its replica. It should be unstable structurally and chemically. It should provide the scope for slow changes that are required for evolution.
Option C, ma'am.
It should be unstable structurally
Option
and
C, ma'am.
chemically, ma'am.
E, ma'am.
Ramya, Madhumita, Saundarya, Kanchana, Saundarya and Soumya answered through mic. Kavita, Varsha, Gayatri, Lakshmi, Parvati, Chaitra, Jyoti, Prema, Kumar, Viresh. Okay. Option C, it should be unstable structurally and chemically is the right answer. Now let us brush up certain concepts again. Okay. So this is regarding the double helix DNA model composed of nucleotide, sugar, phosphate bases, bases AG, adenine and guanine, and pyrimidines, thymine and cytosine. Chargaff's discovery, we should remember percentage of A is equal to percentage of B. A will be bonded with T, always double bond, C with triple bond with G. Then sugar ಮತ್ತೆ phosphate ಅನ್ನೋದು DNA backbones ಆಗ್ತವೆ. They will be held together by covalent bond. Sugar ಮತ್ತೆ phosphate covalent bond ಇಂದ held ಆಗಿರ್ತವೆ. ಮತ್ತೆ those that bond will be anti-parallel. Interior bases ಏನಿರ್ತವೆ, nitrogenous bases, those are hydrophobic, then held together by hydrogen bonds. Sugar phosphate covalent bonds, whereas bases will be held by hydrogen bonds. Purine must always pair with the pyrimidine, never purine or pyrimidine. Andre pyrimidine will not link with pyrimidine and purine will not link with purine. Then semi-conservative replication. Gayatri, can you read out the slide, please?
Semi-conservative replication. Base pairing allows existing DNA strand to serve as templates for new complementary strands. Each strand of DNA is half old and half new.
Okay, thank you. Now, okay. Saundarya R. Read out this first one. A is there, no this one. Diagram ಕೆಳಗಡೆ ಕಾಣಿಸ್ತಾ ಇದೆನಾ? Whether the font is visible?
Parent. The parent molecule has two complementary strands of DNA. Each base is paired by hydrogen bonding with its specific partner, A with T and G with C.
Okay, thank you. Now I request Saundarya ಆಯ್ತಲ್ವಾ? Hemashri, can you read out second one?
Yes, the first step in replication is separation of the two DNA strands.
Okay. Now, Kanchana Raj, can you tell me which enzyme is used here to separate two DNA strands?
Ma'am, I can't understand.
Semi-conservative method replication do very important question for theory alva.
Yes, ma'am.
The first step in replication is separation of the two DNA strands. Idralli yav enzyme use agutte? Very good. Preeti, Lakshmi,
Helicase enzyme.
enzyme will be used. Very good. Now, Kumar, are you here? Can you read out the third one, please? Kumar? ಯಾರಾದ್ರೂ ನಿಮ್ಗೆ ಓದಿದ್ರೆ ಬಂದು ಹೊಡಿತಾರೇನಪ್ಪ? ಮೈಕ್ ಆನ್ ಮಾಡಿ ಓದ್ಬೇಕಲ್ವಾ?
The first step in replication is separation of two DNA strands.
Third one I said C. Each parental strand.
Each parental strand now serves as a template that determines the order of nucleotides along a new complementary strand.
Thank you. Then, Hemashri, can you read out the fourth one?
Yes, ma'am. The nucleotides are connected to form the sugar phosphate backbones of the new strands. Each daughter DNA molecule consists of one parental strand and one new strand.
Okay. Lohit, can you tell me which bond is present between the sugar and phosphate? Lohit,
Yes, ma'am.
which bond is present between sugar and phosphate?
Covalent bond, ma'am.
Covalent bond. Tiffin ಮಾಡ್ತಾ ಇದ್ದೇನೋ?
Yes, sir.
Okay, Messelson and Stan labeled DNA with heavy isotope 15 Newton. Any new DNA contained 14 Newton. When results were centrifuged, there was 15N and 14N hybrid, which confirmed the semi-conservative model. So this is a high level. Okay, fine. Now, just a minute. Replication force catalyze. Okay, DNA polymerase function, it catalyzes the elongation of the replication force. This we will read later. We have some questions. Either mail is all certain questions. Idana solve ಮಾಡಿ ಈ concepts ಅನ್ನ ಓದೋಣ. ಒಂದು ನಿಮಿಷ. Okay. Semi-conservative method was first demonstrated in option A, Drosophila melanogaster, option B, Escherichia coli, option C, Streptococcus pneumoniae, option D, Salmonella typhimurium.
Option B, ma'am. Escherichia coli.
Kavita, Rahul, Oppo A16, option C, Saumyanara, Lakshmi, Madhumita, option C, Aishwarya Kumar, option B, Hemashri, Veeresh, Samar, option C. Okay, Soundarya, option C. Nuhu Sayyed, Parvati, option B. So most of them having confusion between option B and option C. Please mute yourself. Whose mic is switched on, please mute yourself. Okay, so the right answer is option D, that is Escherichia coli. Semi-conservative replication of DNA was first demonstrated by Matthew ಅನ್ನೋರು semi-conservative replication ನ first demonstrate ಮಾಡ್ತಾರೆ. Messelson and Frankenstein in 1958 with the help of E. coli. ಈ ಇಬ್ರು ಸೈಂಟಿಸ್ಟ್ ಅದನ್ನ ಡೆವಲಪ್ ಮಾಡ್ತಾರೆ. ಯಾರು ಮ್ಯಾಥ್ಯೂ ಡಿಸ್ಕವರಿ ಮಾಡೋದನ್ನ they develop with the help of E. coli. So the answer is option B, E. coli. Which one of the following is not a criterion of a genetic material? Genetic material, the characteristic alla. Before also you discussed this question, but the options were different. These options ಬೇರೆ ಇದೆ. So option A, should not provide the scope for changes for evolution, should be able to express itself in the form of Mendelian character. Options are jumbled, that's it. Same question. Should be able to generate
Option A, ma'am.
Option A, ma'am.
Option A, ma'am.
Option A. Most of you answered it as option A.
That provide the scope for changes for evolution. Option A, ma'am.
The process of translation of mRNA to proteins begins as soon as. Until I complete the question, please don't message your answers. Translation of mRNA to proteins ಯಾವಾಗ ಆಗುತ್ತೆ? Option A. The tRNA is activated and the larger subunit of ribosome encounters mRNA. Option B. The small subunit of ribosome encounters mRNA. Option C. The larger subunit of ribosome encounters mRNA. Option D. Both the subunits join together to bind with mRNA. What is the answer? Chandru Agar voted for option A. Kumar, option D. Aishwarya, option A. Lohit, option D. B. Okay. Kavita, option A.
Option 2, ma'am. The small subunit of ribosome encounters mRNA. Option B, ma'am.
Kavita, option B. Veeresh, option A. Jyoti, option B. So there is a fight between option A and option B. Most of them voted for option B. Let us check. The right answer here is option B, that is the small subunit of ribosome encounters mRNA. It is right answer. The next one is The process of translation of mRNA to proteins synthesis begins as soon as smaller subunit of ribosomes encounters mRNA. Anticodon occurs in option option A tRNA, option B mRNA, option C rRNA, option D DNA.
Option A, ma'am, tRNA.
Option A, ma'am, tRNA.
Pavitra, Varsha, Ramya, Veeresh, Sangeeta, Parvati, Lakshmi, VM, Dalbanchan, Chaitra. Okay. Most of you voted for option A and most of you answered through mic as option A. Option A, tRNA is a right answer. And anticodon is a trinucleotide sequence. Anticodon ಏನು trinucleotide. ಮೂರು nucleotide ಇರುತ್ತೆ which located at one end of tRNA which is complementary to a corresponding codon in mRNA. Next question. Variations of proteins are due to option A sequence of amino acids, option B number of amino acids, option C R group, option D none.
Option D, ma'am.
Chandru option A, Kumar option D, Oppo is 16 option A, Gayatri option E, Veeresh, Sinchana, Kavita, Saundarya option D, Saumya S, Harshita option A, Lohit option A, Veeresh option A. Okay, most of you are answering option A. Few of them are selecting option D. So let us check the answer. Okay, fine. Kanchana Raj, next. The right answer is option A, that is sequence of amino acids. Okay, now Vidya, can you read out the slide? Vidya, are you here?
Yes, ma'am.
Yeah, please read out the slide.
Proteins are polymers of amino acids. A number of amino acids are joined together by peptide bonds to form proteins. The sequence of amino acids is decided by the sequence of nucleotides in DNA. The sequence of nucleotides in DNA determines the sequence of nucleotides in mRNA. The sequence of nucleotides in mRNA decides the sequence of amino acids in the protein.
Okay, thank you. So option A is the right answer. Now let us discuss some questions. Brush up some ideas now. Okay. So DNA polymerase, there are 11 different DNA polymerase in eukaryotes. DNA is antiparallel with 5' to 3', which is running in opposite direction. 5' endally free phosphate group irutte, 3' endally free OH group irutte. So e OH group annodu, it will be attached to the deoxyribose. It may be asked, 3' end of DNA polymerase will be attached to deoxyribose in DNA. New nucleotides are always added to the 3' end by a DNA polymerase 3. So DNA polymerase 3 helps in addition of new nucleotides. Yav endige 3' endige. Then, so this is a diagrammatic representation. Okay. Leading versus lagging strand, very important concept. So leading strand andre DNA polymerase 3 moves with the replication fork. DNA polymerase 3 en madutte, moves with the replication this side. Okay, it moves with the replication fork, synthesizing new complementary strand. Idenide alva, idu new complementary strand agutte of DNA. Lagging strand enide, this is a lagging strand which you are observing in the diagram. DNA polymerase 3 moves away ಅಂದ್ರೆ ಅದರಿಂದ ದೂರ ಹೋಗುತ್ತೆ, or against the replication fork. It moves opposite to the replication fork and therefore synthesize fragments. Aden madutte, it synthesize or it produces fragments of a new complementary strand of DNA, which are called as Okazaki fragments. So DNA ligase annodu, it joins Okazaki fragments together later. DNA ligase. Then we have regarding the primers. Who wants to read regarding primers? Please.
DNA polymerase 3 cannot start the process of polynucleotide synthesis, can only add nucleotides to existing chain. Primers start the initial chain, 5 to 10 nucleotides long, can be RNA or DNA. Primase An RNA polymerase starts in the primer. DNA polymerase 1 removes primers and replaces with DNA after a new strand has been created.
Thank you. So ಇಲ್ಲಿ DNA polymerase ಅನ್ನೋದು ಅದು ಸ್ಟಾರ್ಟ್ ಮಾಡಲ್ಲ ಪ್ರೊಸೆಸ್ ಅನ್ನು. It can only add the new nucleotides, whereas the things which start the process is primers, which starts the initial chain. So primers ಹೇಗಿರ್ತವೆ? These are made up of 5 to 10 nucleotides and they can be RNA or DNA. Yes, Samat, you are right. Then DNA polymerase 1 removes primer and replaces with DNA. Polymerase 1 function in ಮಾಡುತ್ತೆ ಅಂದ್ರೆ it removes the primer and replaces the primer with the DNA once the new complementary strand has been created. This is regarding primers. And this is regarding the function of certain enzymes, which we will read later. Now we will discuss some questions again. Okay. Complete the flow chart on central dogma. Observe carefully. Don't answer in a hurry. Option 1, A, replication, B, transcription, C, translation, D, protein. A, transcription, B, translation, C, replication, D, protein. A, replication, B, transcription, C, transduction, D, protein. A, transcription, B, replication, C, transcription, D, transduction. Read out the question properly and then answer.
Option A, ma'am. Replication,
Option A, ma'am.
transcription, translation, and protein.
Option A, ma'am.
Okay, Kachanaraj, Saundarya, and Lohit answered to mine, and I got 12
Option A, ma'am.
or chat answers. Most of them are voting for A. Very easy question, very quick responses. Option 1, that is A, replication agate. After that, transcription occurs where DNA will be converted into mRNA, and then translation occurs. Okay, then mRNA to protein, then last protein will be formed. So option A is the right answer. Very good. Fine. What is the role of RNA polymerase 3 in the process of transcription in eukaryotes? Eukaryotes ಅಲ್ಲಿ RNA polymerase 3 the function ಅಥವಾ ರೋಲ್ ಏನು ಅಂತ ಕೇಳಿದ್ದಾರೆ. Option A, transcribes precursor of mRNA. Option B, transcribes only snRNAs. Option C, transcribes only rRNAs. Option C, transcribes 3RNA, 5sRNA, snRNA.
ಮ್ಯಾಮ್ ಆಪ್ಷನ್ ಡಿ ಟ್ರಾನ್ಸ್ಕ್ರೈಬ್ಸ್ ಟಿಆರ್ಎನ್ಎ ಐಆರ್ಎನ್ಎ ಮತ್ತೆ ಎಸ್ಎನ್ಆರ್ಎನ್ಎ.
Saundarya, Soumya, Chaitra, Nuhu Sayyed, Madhumita, Sangeeta, most of them voted for option D. Okay, Asha, Dyuti, Veeresh, Prema, Chaitra, Aishwarya, Pavitra, Parvati. Okay, most of them answering for option D, that is transcribes tRNA, 5sRNA and snRNA. It is a right answer. Very good. Then column 1 ನಲ್ಲಿ ಸೈಟ್ ಅನ್ನ ಕೊಟ್ಟಿದ್ದಾರೆ. ಕಾಲಮ್ 2 ಅಲ್ಲಿ ಅಲ್ಲಿ ಏನಾಗುತ್ತೆ ಅನ್ನೋದನ್ನ ಕೊಟ್ಟಿದ್ದಾರೆ. ವಾಟ್ ಹ್ಯಾಪನ್ಸ್ ದೇರ್. ಸೊ ದ ಕರೆಕ್ಟ್ ಮ್ಯಾಚ್ ಯು ಶುಡ್ ಫೈಂಡ್. ಆಪ್ಷನ್ 1 ಅಲ್ಲಿ ನೋಡಿ ಮ್ಯಾಚಿಂಗ್ ಪೇರ್ ಕೊಟ್ಟಿದ್ದಾರೆ. ಐ ಕಾಂಟ್ ಸೇ ಇಟ್ because first one is B, second one is A, third one is C, fourth one is D. You only read out the question and then answer. Read column 1 and column 2. Soumya Naral answered through chat. Okay.
Option C, ma'am.
Saundarya option C. Varsha option D. Nuhu Sayyed option D.
A, ma'am.
A. Okay. Hemashri option D. Madhumita option D. Veeresh option D. Chandru Vaggar option A. Let us see who is right. Option B. So ಇಲ್ಲಿ ಏನಾಗುತ್ತೆ operator site will be option B. First one B. Other operator site B, binding site. Promoter site, one ನಿಮಿಷ, binding site, yeah, for RNA polymerase. Then structural G, T, code for repressor molecule. Then fourth one C, that is codes for enzymoprotein. Then next question. What is the chemical name for thymine known as? Very easy question. 5-methoxyuracil, B 3-methoxyuracil, C 5-methyl-uracil, D 3-methyl-uracil.
Option C,
Option
ma'am.
A, ma'am, 5-methoxyuracil.
Rohit, Sumant, Pavitra, Kanchana Raj, Nuhu Sayyed, Rajeshwari, Ramya, Hemashri, Sinchana, Lakshmi Vadiyar, Vanita. Most of them voted for option A, 5-methoxyuracil. 5-methoxyuracil. 5-methyl-uracil.
No, ma'am. Methyl uracil.
It is 5-methoxy alla 5-methyl-uracil. C ಇರ್ಬೇಕು. C ಅದು.
Yes, ma'am. Option C, ma'am. Right answer. Option C, ma'am.
5-methyl, methoxy, 3-methyl. 5-methyl-uracil.
Uracil.
Okay, good. Thank you. 21st question. Down syndrome in humans is due to 3X chromosome, 3 copies of chromosome 21, C monosomy, D 2Y chromosome.
Option B, ma'am.
Option B, ma'am. Option B, ma'am.
Option
B.
3 copies of chromosome 21. Option B, ma'am.
Option B, ma'am.
Option B, 3 copies of chromosome 21 is a right answer. Now let us brush up certain concepts. I'm sorry. Okay. So there are certain enzymes which are involved in DNA replication. Helicase, it untwists or unwinds the DNA and creates replication fork. It breaks the hydrogen bonds between the bases. Then topoisomerase annodhenagate relieves tighter twisting of the DNA, coiled DNA, and existed twisted DNA caused by the helix. Single strand binding protein it stabilizes unpaired DNA strands. It stabilizes it until new complementary strand will be added. Until that it should be stable. No. So single strand binding protein ಅದಕ್ಕೆ help ಮಾಡುತ್ತೆ. Keeps double helix from rewinding. Then, okay. Proofreading and repair. DNA polymerase self-corrects mismatched base pairs, which is called mismatch repair. DNA polymerase self-correction ಮಾಡ್ಕೊಳ್ಳುತ್ತೆ. The next one is nuclease. It cuts out bad or mutated DNA errors. Mutated DNA errors will be deleted by nuclease. So mutated DNA errors ಯಾವಾಗ ಆಗ್ತವೆ? It occurs 1 in a billion. DNA polymerase and DNA ligase act the correct DNA nucleotide excision repair. Fine. Next one, just a minute. The next one is regarding the telomeres. Okay. Soumya, can you read it?
Telomere of DNA sequences. TTAGGG is a telomere in humans. 100 to 1000 repetitions protect the DNA strand shortened with each round of DNA replication. Somatic cells contributes to tagging. Telomeres added.
Telomerase adds telomeres to gametes. Thank you. I'm sorry. Okay. Now, next question. Read the following 4 statements. Just a minute. A to D. Then how many of the above statements are right? In transcription, adenosine pairs with uracil. Option statement B, regulation of lac operon by repressor is referred to as positive regulation. Human genome has approximately 50,000 genes. Hemophilia is a sex-linked recessive disease. How many of the above statements are right? Option A 2, option B 3, option C 4, option D 1.
Option
D, ma'am.
Option D
Option
1.
D, ma'am.
Which statement is right? Can anyone tell me? ಯಾವ statement correct?
Recessive disease, ma'am.
Okay. What about
the
ABC?
hemophilia is
Option B,
sex
ma'am.
linked disease.
Okay, that is the right statement. What about statements ABC? Wrong idena? Okay. Statement A. Transcription is the process of formation of mRNA from DNA. So option A is the right answer. ಇಲ್ಲಿ statement ಅನ್ನ ಕೊಟ್ಟಿದ್ದಾರೆ. So statement ಏನ್ ಹೇಳುತ್ತೆ? Transcription is the process of formation of mRNA from DNA ಅಂತ ಹೇಳುತ್ತೆ. Statement B. The repressor protein binds to the operator region. Two statements correct ಇದೆ ಅಂತ ಕೊಟ್ಟಿದ್ದಾರೆ. So the regulation of operon by the repressor is called negative regulation. So illénide? Positive regulation. So this is a wrong statement, alva? Then the human genome ಅಲ್ಲಿ 30,000 ಇದೆ. ಇಲ್ಲಿ ಏನ್ ಕೊಟ್ಟಿದ್ದಾರೆ? 50,000 ಅಂತ ಕೊಟ್ಟಿದ್ದಾರೆ. ಹೌದಾ? Then hemophilia is a X-linked recessive disease. So statement A ಮತ್ತೆ statement D ಎರಡು correct ಆಗಿದೆ. So option A, that is two statements are correct. Option A, two statements are correct. That is the right answer. Option A is the right answer. Okay. Some of them ಏನ್ ಮಾಡ್ತಾರೆ ಗೊತ್ತಾ? ಇದನ್ನ ನೋಡ್ತಾರೆ. A, D ಇದಷ್ಟೇ ನೋಡ್ತಾರೆ. Statements ಅನ್ನ ನೋಡಿ this option you should select. Okay. While selecting in the exam, you guys will be confused. So be careful while answering. Next, select the incorrect statement from the following. A, B, C, D ಅಂತ ಅನ್ಕೊಳ್ಳೋಣ. A statement ಏನ್ ಹೇಳುತ್ತೆ? Galactosemia is an inborn error of metabolism. B statement, small population size results in random genetic drift in population. C statement, baldness is a sex-limited trait. D statement, linkage is an exception to the principle of independent assortment in heredity. ಇದರಲ್ಲಿ ಯಾವ ಸ್ಟೇಟ್ಮೆಂಟ್ ರಾಂಗ್ ಇದೆ? A, B, C, D. Incorrect, which is wrong. Samarth answered as option B. Gayatri voted for option A. What about others? Dyuti option D. Then Veeresh option B. Lohit Kumar option C. Gayatri, Pavitra, Kavita, Tanchana Raj option C. S. Harshita option C. Veeresh, Saumyanara, Prema option C. Okay. Option C, baldness is a sex-limited trait. Option C is the right answer. As you can see here, galactosemia is an inborn error of metabolism characterized by the inability of galactose. It's correct agide. The effect of genetic drift on a small population is large as compared to that in larger population. This statement is also right. Then we have baldness is an example of sex-influenced trait which is governed by autosomal genes. So ಇಲ್ಲಿ ಏನ್ ಕೊಟ್ಟಿದ್ದಾರೆ? sex-limited trait ಅಂತ ಕೊಟ್ಟಿದ್ದಾರೆ. As they are controlled by autosomal genes. Option C is the incorrect statement. Now, just a minute. Okay, fine. I'm sorry. Sickle cell anemia is caused by substitution of valine by glutamic acid in the beta globin chain, caused by change in a single base pair of DNA, characterized by the elongated sickle-like RBCs with the nucleus, and autosomal linked dominant trait.
Option A, ma'am. Caused by substitution of
Voted for option A, that is substitution of valine by glutamic acid in the beta globin chain. So option A is the right answer. Which among the following is the exact ratio of guanine to cytosine in a DNA double helical structure?
Option A, ma'am.
2 is
Option
to 1.
A, ma'am. 3:2.
C is 1 is to 1.
Option A, ma'am. 3:2.
Option A, Veeresh, Hemashri, Dyuti, Sinchana, most of them voted for option A.
Option A, ma'am.
The right answer, that is option A, 3:2. Now, let us proceed with the new thing of today. So as I said, we are going to start 5-star bio from today. last 5 to 10 minutes we are going to discuss on that. 5-star bio ಅಂತ ಅಂದ್ರೆ ಏನು, ನಿನ್ನೆ ಮಾಡಿರೋ ಕಾನ್ಸೆಪ್ಟ್ ಅಥವಾ ಇವತ್ತೇ ಮಾಡಿರೋ ಕಾನ್ಸೆಪ್ಟ್ ಮೇಲೆ 5 ಕ್ವಶನ್ಸ್ ನಾನು ಸೆಲೆಕ್ಟ್ ಮಾಡಿರ್ತೀನಿ. So that 5 questions you should answer in 5 seconds. Mic should not be used. I repeat, mic ಅನ್ನ ಯೂಸ್ ಮಾಡೋ ಹಾಗಿಲ್ಲ, only should be answered through chat. Then 5 seconds ಅಲ್ಲಿ ಹೇಳ್ಬೇಕು. I will be counting 5 4 3 2 1. ನಾನು ಕ್ವಶನ್ ಓದಿ ಆಪ್ಷನ್ಸ್ ಓದಿ when I start to begin the count you have to answer. ಕ್ವಶನ್ ಕೇಳಿದ ತಕ್ಷಣ ಆನ್ಸರ್ ಮಾಡೋದಲ್ಲ. Those answers will not be considered. Okay. Then after that, okay, the answer should be message in the matra ಮಾಡಬೇಕು. I will be opening that. Today we are going to discuss 5-star bio on the topic. This one, 5-star bio ಯಾವುದು, principle of inheritance and variation ಬಗ್ಗೆ. Okay, be ready with your chat box, with your keyboard. And these questions, whatever these questions are there, no, e questions anna nanu I will be collecting these questions from the famous publications or from the famous books which are available in the market. ನೀವು ಎಲ್ಲ books ಅನ್ನ ತಗೊಂಡು ರೆಫರ್ ಮಾಡಕ್ ಕಷ್ಟ ಆಗುತ್ತೆ. So I will be doing it and I will be presenting the questions from those publications and the books. So let us proceed to the 5-star bio today. Be ready guys and be active too.
Yes, ma'am.
Yeah. ಒಂದ್ 2 ನಿಮಿಷ ನನಗೆ ಯಾವುದೇ ಮೆಸೇಜ್ ಬರಬಾರ್ದು ಚಾಟ್ ಬಾಕ್ಸ್ ಅಲ್ಲಿ. Then I will start. Okay. 5-star bio starts now. The first question of today is principle of inheritance and variance. The first question is, among the 7 pairs of contrasting characters in pea plant as studied by Mendel, the number of traits related to flower, pod and seed respectively are option A 222, option B 221, option C 122, option D 112. Your time starts now. I'm opening the chat box. Option A 222, 5 4 3 2 1. Okay, the first message I got from Varsha Kanaki, but the answer is wrong. Okay. Option A is the right answer. That is 222. No one wins here. Okay. Now I will open the chat box and keep just a minute. Okay. So in honeybees, females are having dash and males are having dash chromosome. Read out the options and start answering. A, B, C, D. Okay. Your time starts now. Chandru answered it correctly. Chandru Waggar. Option C, that is diploid 32, haploid 16 anta. So answer is option C is the right answer. The next question goes like this. A man having the genotype EEFFGGHH can produce P number of genetically different sperms, and women of genotype LILLMMNN can generate Q number of genetically different eggs. Determine the values of P and Q. Don't answer now until I read out the option. A, Samarth, your answer is not considered. A, P is equal to 4, Q is equal to 4. B, P is equal to 4, Q is equal to 8. C, P is equal to 8, Q is equal to 4. D, P is equal to 8, Q is equal to 8. Your time starts now. 5, 4, 3, 2, 1. The first correct answer is messaged by Hemashri. That is option B. Option B. P is equal to 4, Q is equal to 8 is the right answer. So when nan chat box alli first answer nan 5 4 3 2 1 ಆದ್ಮೇಲೆ ಯಾರ್ ಆನ್ಸರ್ ಬರುತ್ತಲ್ಲ ಅದನ್ನ ನಾನು ಹೇಳ್ತೀನಿ. Most of you are answering prior only. So those answers I will not consider. Now here ಇದನ್ನ ಏನ್ ಮಾಡ್ಬೇಕು ಅಂದ್ರೆ, how to do this I will just show you. 2 raised to n. N is nothing but number of heterozygous condition. Male ali heterozygous ಯಾವ್ಯಾವಿವೆ? F ಇದೆ ಮತ್ತೆ G ಇದೆ. So total 2 heterozygous condition, heterozygosity is there. 2 raised to 2, that is equal to 4. 4 different genetically different gametes will be formed. Same way, women ali we have 3 heterozygosity. So women ali 2 raised to 3, that will be 8. So 8 genetically different gametes will be formed in case of women. So answer is option B is a right answer. Okay, fine. Now, fourth one, you don't have option. You have to answer. The gene disorder phenylketonuria is an example for dash. 5, 4, 3, 2, 1. Chaitra answered it correctly. That is pleiotropy. The first answer is given by Chaitra. Pleiotropy is a right answer. Last question of 5-star bio today. How many types of gametes can be produced by a diploid organism who is heterozygous for 4 loci? Who is heterozygous for 4 loci? Option A 4, option B 8, option C 16, option D 32. Option C 16 is the right answer. Who answered it first? 16, 16, 16. Okay, Angel answered first. It has 16. Option C, that is 16 is the right answer. So this is about 5-star bio today regarding principle of variance and inheritance. I hope everyone enjoyed this 5-star bio.
Yes, ma'am.
Ma'am.
Okay,
Yes.
then let us continue this or no?
Continue, ma'am.
Yes, ma'am.
All of you enjoyed?
Yes,
Yes,
ma'am.
ma'am.
Okay, then let us end up the session here. Have a nice day. Thank you.
Thank you, ma'am.